Hi everyone and also to you Mama mismo. I am Riza Cañado. We are a group 2. In this video, we'll be talking about plant acyl CoA binding proteins, an emerging family involved in plant development and stress responses. A group of proteins that help plants grow and respond to stress. Before that, let me introduce my group members. Chanika Galela, Angeline Fernandez, Ivory Cartala, Jason Talian, and Jisma Arqueta. Introductions. ECL-CoA binding proteins, ECBPS, are conserved family of proteins found across eukaryotes, playing essential roles in lipid metabolisms by binding medium to long-chain acyl-CoA esters. In plants, ECBPS are more complex and diverse, indicating involved functions specific to plant physiology. Recent studies have uncovered their involvement not just in basic lipid transport but also in critical biological processes like embryogenesis, organ development and defense, mechanisms against abiotic through salinity and cold, abiotic pathogens, stress. The increasing ability of genetic and molecular tools has facilitated deeper investigations. This type of image is called a flowchart or concept diagram. It visually represents key points and relationships in case summarizing the roles of ECVPS in plants.
Your name is Moira. I am Nelly Cinderella from Methodology and Materials. As a literature review, this article synthesizes findings from various experimental research papers. The methods involved first is bioinformatic analysis. Classification of plant exhibits into four classes based on domain structures using protein databases. Second, subcellular localization. Comparative studies using GFP fusion proteins and on-focal microscopy to determine where HMP is localized within cells. Third, gene expression profiling, analysis of transcriptomic data and expression patterns under stress using RT-PCR microarrays and promoter US ocean studies. Four is mutant and overexpression analysis, insight from Arabidopsis and rice mutants or transgenic plants overexpressing ECBPs. Fifth, protein interaction studies, co-immunoprecipitation yes, two hybrid assays, and all-down assays to understand interaction networks. This methodology allows a comprehensive understanding of how ECBPs function at the molecular, cellular, and physiological levels.
As a literature review, this article synthesizes finding various experimental research papers. This includes the bioinformatic analysis, the capitalizations of plant ACBPS based on the four classes of the domain structure using the databases of the proteins. Then second is the subcellular localizations. Comparative studies using the GFP-TAG fusion studies and confocal microscopy to determine where ECVP is localized within the cell. And then third is the gene expression profiling, analysis of transchromatic data and expression patterns using the RT-PCR, the microarrays, and the promoter, which is the GUS studies. And then last is the protein interaction studies. Co-immunoprecipitations, yes, two hybrid assays, and then the assays fall down to understand the interaction network. This methodology allows a comprehensive understanding to determine how ACBPS function on molecular, cellular, and the psychological levels.
Subcellular localization. Different classes show varied localization. Class 1, cytosol. Class 2, plasma membrane. Class 3 and 4, multiple organelles, plasma membrane, and haploplast. Explanation. Class 1, cytosol. These proteins are found in the cytosol, the fluid portion inside the cell where various metabolic processes occur. It allows quick interaction with other cellular components. Class 2, plasma membrane These proteins localize to the plasma membrane and endoplasmic reticulum The plasma membrane controls entry, exit of substances, while the is involved in the protein folding and transport. Class 3 and 4, multiple organelles, plasma membrane, and haploplast. These proteins are more versatile, targeting multiple locations. The and plasma membrane, as well as the haploplast, the space outside the cell membrane. This suggests complex roles in transport, signing, or defense.
Roles in plant development. CBP6 class 1 is involved in seed germinations and lipid homeostasis. CBP4 and CBP5 affect embryo and organ development. CBPS regulate cuticle and formation, floral development, and leaf size. CBP6 class 1 plays a key role in seed germinations and maintaining lipid balance, which is crucial for energy and membranes in structure during early growth. CBP4 and CBP5, these proteins influence embryo formations and organ development, impacting how the plant's basic structures takes a shape. General ACBP functions and ACBP also help regulate cuticles and formation, protective layer, floral development, and leaf size, all important for plant growth and reproductions.
Fourth, roles in stress responses. Abiotic stress. ACBPs like ACBP1 and ACBP2 responds to cold, drought, and heavy metals, modulating membranes, lipids, compositions, and signaling. So it means that is abiotic stress. ACBPs such as CBP1 and CBP2 help plant responds to cold, drought, and heavy metal stress by adjusting membranes, lipids, and stress signaling pathways. Biotech stress. ACBPs modulate defense responses via interactions with pathogens-related pathways, e.g. NPR1 in salicylic acid signaling. So here ACBPs are involved in plant defense interacting with proteins like NPR1 in the salicylic acid pathway to enhance resistance against pathogens. Overexpression of certain ACBPs improves tolerance to cadmium and oxidative stress, while here it involves in stress tolerance. That means overexpression some ACBPs can boost plant tolerance to cadmium and oxidative stress, showing their protective role.
Number 5 is interaction with signaling molecules. So they have two. First is ACBPS interact with lipids like phosphatidic acid and oxalipids, initiating stress-related signaling. So ACBPS or acyl-CoA binding proteins bind to certain lipids that act as signals during stress, helping the plant respond by activating defense pathways. Second is some ACBPS are involved in vesicle trafficking and membranes remodeling under stress. During stress, ACBPS help transport cellular materials in small vesicles and access in reshaping membranes to maintain cell stability and function.
So, conclusion. Plant ECBPs are versatile proteins critical for lipid transport and regulation, but also serve broader roles in development and stress adaptation. Their diverse structure and localization enable them to interact with various partners and pathways. This review emphasizes the need for functional studies in crops, understanding post-translational modification of ECBPs, exploring their potential in agricultural biotechnology. Example is engineering stress-tolerant crops. ECBPs represent promising targets for improving plant resilience and productivity under changing environmental conditions.